In 1989, on a dairy farm in Colorado, a 28-year-old man climbed down into a pit, a manure pit, about 20 feet by 24 feet, 10 feet deep. He needed to fix a pin on an agitator shaft. Simple job. He climbed down, and he collapsed. His brother saw him fall. The brother jumped in after him. He collapsed too. Their father, 65 years old, saw both his sons go down. He went in. A nephew followed. Then a 15-year-old grandson. 5 members of the same family. Dead, one after another, in a pit where the air looked perfectly clear. No smoke, no fire, no visible threat of any kind. Just air that was not air anymore. What killed them was not a disease, not a chemical spill, not a weapon. It was a gas that comes from rotting things. And here is what I want you to sit with for a moment. Every living thing that has ever died, every animal, every person, every creature with proteins in its body has produced this gas during its decay. Your body will produce it too. The question is not whether it happens. The question is how a colorless, familiar substance, something your own cells manufacture right now while you are alive and breathing, becomes a killing machine after death. Let me start with what happens the moment your heart stops, because that is where the chemistry begins, and the chemistry is beautiful, terrible but beautiful. When the heart stops pumping blood, oxygen delivery to your cells ends. Within minutes, cells that were humming along, doing their jobs, processing fuel, taking out garbage, suddenly find themselves in the dark. The little power plants inside each cell, the mitochondria, they shut down. And without power, the cell cannot maintain its own boundaries. The membranes that held everything in place start leaking. Enzymes that were carefully stored in compartments, kept away from the delicate structures they could damage, those enzymes spill out. They start digesting the very cell that made them. This is called autolysis. Self-digestion, and it is not bacteria doing it, not yet. This is your own machinery turning on itself. Think of it like a factory where the power goes out. The assembly lines stop, but the chemicals stored in barrels are still reactive. Without the systems that kept them contained, they start eating through the floors. The liver goes first because the liver is packed with enzymes. Proteases, lipases, nucleases, all neatly stored in membrane-bound packages called lysosomes. While you were alive, those membranes held. They kept the digestive enzymes locked away from the surrounding tissue like acid in a bottle. When the cell dies and the membrane breaks down, those enzymes spill out and go to work on everything they touch. The brain follows quickly because it is mostly water and fat, extraordinarily soft, extraordinarily vulnerable. Your kidneys, your pancreas, your intestinal lining, they all start coming apart at the molecular level. Meanwhile, damaged blood cells leak out of disintegrating blood vessels and, pulled by gravity, settle to the lowest parts of the body. This is what coroners call livor mortis, those dark reddish-purple patches on the skin of a corpse. It is just blood pooling where gravity takes it, staining the tissue like wine soaking into a tablecloth. And as carbon dioxide accumulates from all this uncontrolled chemistry, the pH drops. The environment inside the body becomes increasingly acidic. Somewhere around this point, a very specific shift happens. The conditions move from favoring aerobic chemistry, the kind that needs oxygen, to anaerobic chemistry, the kind that thrives without it. This shift is the starting gun for what comes next. Because the body is now a paradise for a very specific kind of microbe. You have been carrying these microbes your entire life. They live in your gut, in your intestines, by the trillions. While you were alive, your immune system kept them in check. It said, you stay there, you do your job, you do not cross this line. The moment the immune system goes silent, there is nobody left to enforce the rules. The bacteria do what bacteria have always done. They eat. They start with the intestinal walls. Then they push outward, hitching rides through your blood vessels, spreading to every organ. And as they feast on your proteins, your fats, your carbohydrates, they produce waste products. Gas. Enormous volumes of gas. Now most people know about the bloating. A body swells during decomposition. It can double in size. That bloating is from the gas these bacteria produce. Methane, carbon dioxide, ammonia, nitrogen. But tucked in among those gases is something far more sinister. Something that does not merely smell bad. Something that kills. Hydrogen sulfide. Let me tell you about this molecule because it has a strange double life. 2 atoms of hydrogen, 1 atom of sulfur. That is it. one of the simplest compounds in nature, and your nose knows it instantly. At very low concentrations, hydrogen sulfide smells like rotten eggs, that sulfurous gagging stench that makes you recoil. Oh, everybody has encountered it. A hot spring, a sewer grate on a summer day, a carton of eggs left too long in the back of the refrigerator. But here is the first clue in our mystery, and it is a nasty one. You can smell hydrogen sulfide at concentrations as low as a few parts per billion. Your nose is exquisitely sensitive to it. Evolution made you that way because for millions of years that smell meant danger, rotting food, bad water, disease. Your nose evolved to scream at you, "Get away from this!" So you would think that makes it safe. You would think, well, if I can smell it, I can avoid it. That is what makes hydrogen sulfide one of the most treacherous substances on Earth. Because at higher concentrations, around 120 parts per million, it does something extraordinary. It paralyzes your olfactory nerve. It kills your sense of smell completely in seconds. One moment you smell rotten eggs. The next moment you smell nothing. And most people, when the smell goes away, what do they think? They think it has dissipated. They think the danger has passed. The air smells fine now, but the gas is still there. More of it than before. You just cannot detect it anymore. Your warning system has been disabled by the very thing it was supposed to warn you about. That is diabolical. Not by design, of course. Nature does not design things to be cruel. But the physics and the chemistry conspired to create a perfect trap. Now let me take you inside the cell, because what hydrogen sulfide does at the molecular level is something I find genuinely frightening in its elegance. Every cell in your body runs on a process that is, at bottom, about moving electrons. You eat food. Your digestive system breaks it down into molecules. Those molecules enter your cells. Inside the mitochondria, those little power plants I mentioned, there is a chain of proteins embedded in a membrane. Electrons get passed down this chain like a bucket brigade at a fire. At each step, a tiny bit of energy is captured and used to pump protons across the membrane. Those protons then flow back through a molecular turbine, a rotating enzyme called ATP synthase, and that turbine produces ATP. That is your energy currency. That is what keeps every process in your body running. The very last protein in that electron chain is called cytochrome C oxidase. It sits at the end of the line. Its job is to take the electrons that have been passed all the way down the chain and hand them off to oxygen. That is why you breathe. That is the entire reason you need oxygen. Not to fill your lungs, not to make your blood red, to sit at the end of this electron chain and accept the electrons so the whole process can keep running. Hydrogen sulfide walks right up to cytochrome C oxidase and sits down on it. It binds to the iron and copper atoms at the active site, the very atoms that were supposed to interact with oxygen. And when it does that, the enzyme stops working. The last link in the chain is broken. Electrons pile up with nowhere to go. The proton pumps stall. ATP production crashes. And here is the subtle horror of it. Your cell is still surrounded by oxygen. Oxygen is everywhere. Your lungs are still filling and emptying. If your diaphragm is still working, your blood is still carrying oxygen to every tissue. But the cells cannot use it. It is as if someone plugged the drain in a bathtub and then left the faucet running. The water is right there, the drain is right there, but the connection between them is broken, and everything backs up until the whole system fails. Your brain is the first to notice. It is the most energy-hungry organ in your body, consuming about 20% of your oxygen at rest, despite being only about 2% of your body weight. When ATP production crashes in brain cells, you lose consciousness. Fast. Then the brainstem, the part that controls breathing, goes quiet. Then the heart, another voracious consumer of energy, stops its rhythm. The cascade is terrifyingly swift. This is the same mechanism, by the way, that hydrogen cyanide uses. Cyanide, the legendary poison, the pill in the spy movie, the compound that has haunted the human imagination for centuries. Cyanide kills you by blocking cytochrome C oxidase, and hydrogen sulfide does the same thing. At high concentrations, it is just as lethal. The toxicity of hydrogen sulfide is comparable to that of cyanide. That is not an exaggeration. That is not a metaphor. That is the published chemistry, confirmed by decades of toxicological research. At 500 parts per million, you are in severe danger. At 700 parts per million, you can lose consciousness in 1 or 2 breaths. 1 or 2 breaths. not minutes, breaths. And at 1,000 parts per million, your cardiopulmonary system arrests. Immediately, they call it the knockdown effect in the occupational safety literature. You inhale and you drop. There is no staggering, no gradual decline. Your cells simply stop being able to use oxygen all at once, everywhere in your body. Now bring this back to the dead body. When a person dies, an anaerobic bacteria begin consuming the sulfur-containing amino acids in the tissue, cysteine, methionine. They strip the sulfur atoms out and combine them with hydrogen. Hydrogen sulfide pours out of the decomposing tissue. In an open field, it disperses into the atmosphere. Not pleasant, but not lethal. The concentration never builds high enough. But put that body in an enclosed space, a sealed apartment, a shipping container, a basement, a car, a well, a pit. Now, the gas has nowhere to go. It accumulates, it fills the space, and it is denser than air, so it sinks, it pools at the bottom. Anyone who walks into that space walks into an invisible ocean of poison, and this is where the chain reaction deaths happen. The ones that haunt the safety engineers. A worker goes into a manhole and collapses. His partner sees him fall and jumps in after him. The partner collapses. A third person sees two people down and rushes to help. A fourth, a fifth. In the worst cases, more rescuers die than original victims. There was a case in 2017. 3 workers went into a manhole to investigate why the street above was sagging. The first man descended and was immediately overcome by hydrogen sulfide at lethal concentrations. 2 others went after him and collapsed. When the fire department arrived, one firefighter tried to fit through the opening wearing his breathing apparatus. He could not fit, so he removed it, planning to have his partner hand it down. He took a single breath of the air in that manhole. 1 breath, he was knocked unconscious instantly. He was pulled out and survived. The 3 workers did not. The safety data from the late 1970s and early 1980s showed this pattern explicitly. In confined space incidents surveyed by government researchers, more rescuers died than the people they were trying to save. 60% or more of deaths in some studies were the would-be rescuers. Because the human instinct to help, to rush towards someone in danger, it overrides everything. You see your friend on the ground and your brain does not stop to calculate atmospheric chemistry. It does not ask what the parts per million reading might be. It calculates loyalty, it calculates love, and it sends you down into the same invisible ocean that took the person you are trying to save. There is even a name for it in the research literature: rescue altruism. The psychologists break it down into components: duty of care, identification with the victim, a belief that there is a chance of success even if the risk is enormous, personal courage that overrides rational assessment, all admirable qualities, all potentially fatal in a confined space filled with hydrogen sulfide. Let me pause here and add another layer that makes this even more unsettling, because I have been talking about hydrogen sulfide as a poison, as a killer. But your body is making it right now. Not a lot, tiny amounts, but it is there. Your cells produce hydrogen sulfide as a signaling molecule. Think of it like a whisper between cells. At concentrations in the nanomolar range, vanishingly small, hydrogen sulfide does useful things. It relaxes blood vessels, it helps regulate blood pressure, it plays a role in inflammation, in memory, in protecting cells during stress. There is even research showing that at very low concentrations, hydrogen sulfide donates electrons to the same mitochondrial chain that it poisons at high concentrations. At a whisper, it feeds the machine. At a shout, it destroys it. This is a pattern you see over and over in biology, and I think it tells you something deep about the nature of living systems. The difference between medicine and poison is concentration. The difference between a signaling molecule and a lethal gas is a few orders of magnitude. Water is essential for life, but drink too much too fast and it will kill you. Oxygen keeps you alive, but pure oxygen at high pressure destroys your lungs. Even calcium, the mineral in your bones, will stop your heart if the concentration in your blood rises too high. Nature reuses the same chemistry for opposite purposes, and the only thing that separates life from death is quantity. The knife edge is thin, and your body walks it every moment of every day. The bacteria in your gut produce hydrogen sulfide right now as they digest your food. Sulfate-reducing bacteria, they are called, and they are ancient. They have been doing this work for billions of years, long before animals existed, back when the Earth's atmosphere was very different from today. They break down sulfur-containing compounds, and hydrogen sulfide is one of the waste products. Your body handles it. It has enzymes specifically designed to oxidize sulfide before it reaches dangerous levels. The main pathway runs through an enzyme called sulfide quinone oxidoreductase, right there in the mitochondria, right next to the very enzyme that hydrogen sulfide poisons. It converts the sulfide before it can reach cytochrome C oxidase. There is also a backup system. Recently, researchers discovered that an enzyme called superoxide dismutase, long known for its role in neutralizing reactive oxygen species, also functions as a hydrogen sulfide oxidase. It is another line of defense. Your cells are running a constant detoxification operation against a molecule they also need, a molecule they deliberately produce. But after death, all those detoxification systems shut down. The enzymes denature, the mitochondria collapse, and the bacteria, freed from immune control with an unlimited buffet of sulfur-rich protein, produce hydrogen sulfide on an industrial scale. What was once a whisper becomes a roar. There is another set of molecules I should mention, because they are part of the same story, but often confused with the primary killer: putrescine and cadaverine. The names alone tell you what they are associated with. These are what scientists call biogenic amines, and they come from a different process. When bacteria break down the amino acids, lysine and arginine, they strip off the carboxyl group, the acid part, and what remains are these foul-smelling diamines. Putrescine is a 4-carbon chain with an amine group at each end. Cadaverine has 5 carbons. They are responsible for much of the horrific stench of decomposition. That sweet, cloying, unforgettable smell that anyone who has encountered a decomposing body can never fully forget. But here's what is interesting. Putrescine and cadaverine, despite their gruesome reputation, are not the primary killers. They are toxic at high exposures, yes, but the lethal concentration is far higher than what you would encounter in a typical decomposition scenario. Your body even produces tiny amounts of putrescine during normal cellular metabolism. It plays a role in cell growth. It is not the innocent bystander in this story, but it is not the assassin either. The assassin is the hydrogen sulfide. And there is methane, too. Odorless, colorless, lighter than air. Methane displaces oxygen. A space filled with methane may have perfectly breathable-looking air, but the oxygen fraction has dropped to levels where your brain starves. Normal air has about 21% oxygen. Drop that to 16% and you start feeling dizzy, confused. Drop it to 6% and you can be in a coma within 60 seconds. And methane is explosive. At concentrations between roughly 4.5% and 46% in air, it is flammable. A single spark, a light switch, a static discharge from clothing can ignite it. There have been documented cases where decomposition gases in enclosed spaces were ignited by a spark. A body so thoroughly decomposed that the gas it produced blew up the room. Ammonia is part of the cocktail too. It comes from the breakdown of nitrogen-containing amino acids. It burns the eyes, the throat, the lungs. Carbon dioxide builds up as carbohydrates ferment. Indole and skatole, compounds that come from the bacterial breakdown of the amino acid tryptophan, add their own stench. Skatole is actually the primary odor compound in feces. After death, it is released in quantities that saturate the air. The mixture is complex, and no 2 decomposition scenarios produce exactly the same cocktail. Temperature matters. A hot environment accelerates bacterial growth enormously. The optimal temperature for putrefaction is between about 21 and 38 degrees. Humidity matters. Whether the space is sealed or ventilated matters. The size of the body matters. Even what the person ate in the days before death matters, because it changes the bacterial population in the gut and the available substrates for decomposition. So what do we actually know, putting all of this together? And we know that a dead body is not merely inert matter slowly falling apart. It is an active chemical system. It is a reactor. The bacteria are the workforce, the proteins and fats are the raw material, and the products are a complex cocktail of gases, some foul, some odorless, some irritating, and some lethal. In the open, these gases disperse and the concentrations stay below dangerous thresholds. In an enclosed space, they accumulate, and the most dangerous of them, hydrogen sulfide, is a molecule that disables the very warning system evolution gave you to detect it, and then attacks the same molecular target as one of the most famous poisons in human history. What I want you to carry out of this is not fear. It is respect. Respect for the staggering complexity of chemistry happening everywhere all the time in living things and dead things alike. The same sulfur atom that right now is helping one of your enzymes fold into the right shape, the same sulfur that sits in the amino acid cysteine in your muscles, will someday be liberated by bacteria and recombined into hydrogen sulfide. The atom does not care. It does not know the difference between life and death. It just follows the rules of chemistry. It bonds where the electrons tell it to bond. And there is something profound in that. The gas that kills you is made from you. The poison is not imported from some exotic place. It is assembled from the very atoms that were keeping you alive an hour ago. The sulfur in your proteins, the hydrogen in your water, recombined by organisms that were your passengers, your symbionts, your microscopic tenants who paid their rent by helping you digest your food. And the moment you stop paying attention, the moment the landlord leaves the building, they eat the building. The universe does not waste anything. It does not distinguish between construction and demolition. It has one set of rules, and those rules produce hearts and hurricanes with equal indifference. The atoms in your body were forged in stars, scattered across space by supernova explosions, gathered by gravity into this planet, assembled into amino acids by chemistry we still do not fully understand, folded into proteins, organized into cells, coordinated into a conscious being capable of asking why, and they will be disassembled by the same thermodynamic forces that put them together. Hydrogen sulfide is just one step in that disassembly, a particularly dangerous one, but a step nonetheless in the great recycling project that the universe has been running since the first heavy elements cooled from stellar plasma and began to clump together. If you ever find yourself near a body, in a closed room, a car, a basement, a tank, remember this: if you smell rotten eggs, leave immediately. And if the smell suddenly vanishes, that is not relief. That is the most dangerous signal you will ever receive. That means the concentration has risen past the point where your olfactory nerve can function. Do not go in after someone who has collapsed. Do not be the rescuer who becomes the next victim. Get out, get fresh air. Call for people with proper breathing equipment. The dead do not intend to harm the living, but chemistry does not require intention. It requires only atoms, energy, and time. Tell me this: knowing that your own cells produce the same molecule that kills, that the difference between a signaling molecule and a lethal gas is nothing more than concentration, what does that make you think about the boundary between life and death? Is it as sharp as we pretend, or is it something more like a gradient, a slow fade, where the same chemistry simply tips past a threshold we cannot see?